Hello, welcome everyone to Science Cafe. This is our April session for this event, and we are very pleased to have with us today Dr. Amy Iler, who will be presenting on the title, Drivers and Consequences of Earlier Flowering Under Climate Change. Before we get started, a reminder that this talk will be recorded and available to anyone who can't attend today. So if you've got friends or colleagues who can't attend today, or if you want to relive this experience, the talk will be available on our website. A link will be shared at the end of the talk, but you can also quite easily search for ABG Science Cafe and it should turn up. My name is Loy. I'm the Conservation Ecology Coordinator at the Atlanta Botanical Garden, and I will be your host this evening. At the end of today's talk, we'll have a Q&A session. If y'all have any questions for our speaker today, you are welcome, encouraged even, to ask them. If you look at the bottom of your screen, there is a Q&A button. If you push that button, you can direct your questions there. Okay, let's get started with introductions. Today's speaker, Dr. Amy Iler, graduated from Muskingum University in 2005 with a bachelor's degree in conservation science. She went on to complete a PhD in evolution, ecology, and organismal biology at The Ohio State University in 2010. Her dissertation investigated how an invasive honeysuckle plant affects the pollination of native plants. After this, she started studying the plants and pollinators at the Rocky Mountain Biological Laboratory in Colorado as a postdoc with the University of Maryland. From 2014 to 2016, she continued her research on how climate change affects the timing of biological events as a Marie Curie Research Fellow at the Aarhus Institute of Advanced Studies at Aarhus University in Denmark. In 2016, Amy started a permanent research position at the Chicago Botanic Garden and Northwestern University, where she conducts research, teaches ecology courses, does science outreach, mentors undergraduate, master's, and PhD students. She still returns to the Rocky Mountains of Colorado every summer to study how climate change is affecting plants and pollinators, and serves on the board of trustees for the Rocky Mountain Biological Laboratory. I happen to have the pleasure of knowing Amy in real life. I can attest to Amy's brilliance as a thinker and a researcher. And from firsthand experience working with her and chatting with her students, I know that she's an insightful and very dedicated mentor. If you are or know a scientist of Amy's caliber, you know that simultaneous excellence in science and mentorship doesn't happen by chance. It's a display of prodigious skill, but it's also a willingness and devotion to science and the community. So without further fanboying on my part, please put your virtual hands together for Dr. Amy Iler.
Thank you, Loy. That was such a nice introduction. I'm not sure how to get to my screen now.
Let me see what I can do.
I think you stop sharing and then I can share. There we go.
There we go.
All right, now I will share mine. All right. Thank you so much. So I'm so excited to be here today to tell you all about some of my research. And I will move right on from this slide because you already know the title of my talk. If you haven't noticed, climate change is the thing that's happening and the climate is changing. We have learned quite a bit about the biological responses to climate change. And we can group these into some broad categories, which I will share with you now. So first of all, we see species moving through space to track their preferred climates. And what we're seeing is as the climate's changing, we're seeing species moving up in latitude and up in elevation to track their preferred climate. So now it's warmer, higher up and at higher latitudes, and species are by and large moving up. And this is true of both plants and animals. We're also seeing species move through time to try to track their preferred climates. And this is mostly happening in terms of species moving earlier because now their preferred climates are occurring earlier in the year than they used to. Another thing is that's happening around the world is that species might not move through space or time, and they might just stay and adapt to the new climate. And these are not mutually exclusive events, right? Species can do a combination of all of these things. They can have a range shift, they can move through time, or they could also exhibit adaptations. Unfortunately, if none of these things happen, we might start to see some extinctions in response to climate change as well. But of these things, as you might have guessed from the title of my talk, today I'm going to talk to you about changes through time, and we call that phenology. That just refers to the timing of life cycle events in an organism. So for plants, when they flower, when they leaf out in the spring, which is just now finally happening in Chicago, where I'm based, things like that. For animals, when they migrate. So you'll hear me use the word phenology a lot, and that's what I'm referencing, just the timing of life cycle events. So my work that I'm going to talk to you about today, and most of my research occurs in the Colorado Rocky Mountains in a place called Gothic, Colorado. It's in the western part of the state. And at a place called the Rocky Mountain Biological Laboratory. So this is a field station that is located at 2,900 meters or almost 10,000 feet above sea level. And the reason I work here is because there are some really wonderful long-term records of responses to climate change and of the climate. So if you're studying climate change, it's really handy to have these long-term records. That's something that you'll see as a theme throughout my talk, because the climate does change slowly, right? So it's nice to have this long-term data. Also, we're seeing the climate change happening faster at higher elevations. And this is also true of this area. So it's become kind of a popular spot to study climate change. We get ecologists from all over the world coming here. Now also hydrologists and some other scientists as well. I know you've had other Science Cafe speakers who work here. I won't talk about it too much, but this is where I go here every summer, and I'll call it Rumble for short, RMBL, but it stands for Rocky Mountain Biological Lab. So one of those long-term records that I actually have collected 5 years of the data for as a postdoc allows us to see how the timing of flowering is changing in this location. It's one of the most detailed records of flowering phenology in the whole world, because we go out every other day and count flowers for all the plant species that occur in a series of permanent plots. But what we see, what you can see here on the screen, these are all the plant species listed here on the right, and data points on the left. I'm going to reveal all of them in a minute, but I wanted to orient you first. On the bottom, we have phenological shift or change through time. So anything to the left of a 0 means an event has gone earlier. So for this first species, Claytonia lanceolata, which you probably also have a spring beauty in Georgia, I'm guessing. This is the first thing to flower in the Colorado Rockies, and we see that all of its events are occurring earlier. So first peak and last flowering dates are all flowering earlier than they used to in the past. And if I reveal all of the data points, what I want you to notice is most of those dots fall to the left of the line, meaning that they are indeed getting earlier. But we also see some events like last flowering and some species delaying, and they're flowering for more time than they used to. But what I want to think about today in particular is what is driving all these changes in phenological events. What about the climate is causing these shifts? And so that's the first part of my talk today. We're going to think about what are the underlying drivers of these changes in phenology or timing. And then secondly, we're going to think about the consequences of those changes. So, okay, things are happening earlier. So what? What does this matter for the plants and for their ability to persist as a population? And does this also have effects on how plants interact with one another for pollinators? Okay, so starting off with the first part, what are the drivers of phenological shifts? I'm going to first take you back to the Rockies here to tell you a little bit about how the climate has changed in this location. So we've started, the lab has been measuring temperature since 1974. That's why we're starting at that time point. But what you can see on the left here is that temperatures are increasing in the summer, and the date of snowmelt is getting earlier. So there'll be several figures that I'll show you today where a life cycle event or some kind of phenological event is shown in terms of day of year. And that's just, we've converted a date to a number. So January 1st would be day 1. So for reference here, this regression line is going from June 1st to May 15th. So what we've seen in this location is snowmelt is occurring about 2 weeks earlier than it used to. Why do we care about the timing of snowmelt? It's a very important ecological factor in all high elevation and high latitude ecosystems because it sets the start date for the growing season for all of the herbaceous plants that live there. This is a photo I took in May of 2011 when I was there early to count flowers, but the snow that winter was quite high. So normally we don't have this much snow in May. But it makes a good point that it's a lot of water, right? If all that snow melts, that's a ton of water. And that's another reason why it's important. Ecological factor. It's a pulse of water in the spring that supplies water for a lot of the plant species. And in this ecosystem, then it's pretty dry in terms of precipitation. We don't get much precipitation until July when we get some monsoon rains. And we know from this long-term data set that two factors, both of these factors are associated with earlier flowering. So we see earlier flowering in response to warmer temperatures and in response to earlier snowmelt. However, these two things are correlated with each other, right? So the snow melts earlier when it's warmer. So it's hard to know which one of these things is actually driving the changes in phenology that we're seeing. Is it temperature? Is it snowmelt? Is it both? And to do that, we need to do a field experiment, which is just what one of my master's students did a few years ago. We actually were able to collaborate with a group that already had this experiment set up for another reason, which is another great reason to work at a field station. You can collaborate with people, lots of opportunities for great collaboration. So she used an experiment to tease apart the effects of early snowmelt and warming on flowering phenology. And her work was published last year in a journal called Global Change Biology. And I just want to give her a shout out for that because she did such a great job. So what are the ways that snowmelt might act as a mechanism? The first one is that early snowmelt could lead to earlier heat accumulation. So what do I mean by that? What we know from the literature is that plants are cued by temperature via heat accumulation. So they have a way of tracking how much heat is built up. And once they accumulate so much heat, okay, boom, it's time to flower, or it's time to leaf out. And so what we see when the snow melts out early, shown here by this light blue line, is that the heat can start to accumulate at an earlier date. And in science, we call this growing degree days, abbreviated in short, GDD. So we get more heat accumulated, a larger number of growing degree days when the snowmelt is early compared to when the snowmelt is late. And so in this way, snowmelt might simply be acting as growing degree days in disguise or as temperature in disguise. And maybe that's the only reason, the only way snowmelt's affecting phenology. Another possibility is that early snowmelt leads to an input of soil moisture, which could cue flowering in some way, which we know far less about this as a mechanism, but it could happen in two ways, either directly because the plants obviously are able to sense soil moisture, or via the mobilization of soil nutrients, because when all that snow melts in the spring, that's when the soil nutrients also get mobilized and become available. And so the way we approached this was to manipulate temperature and snowmelt both separately and together. And one way that scientists study climate change out in nature when you don't have any access to electricity to warm something up is with these things called warming chambers. And so these are called open top warming chambers. They're basically like little greenhouses that we put out, and they warm the air temperature 2 degrees Celsius on average. And then the other thing we did was put out black shade cloth when the snow is about 1 meter deep. And because it's black, it causes the snow, it absorbs that heat from the sun, and it causes the snow to melt out earlier. And this results in anywhere from like a 1 to 2 week advancement in the snow melt date. And so we can put these all together. We have warming along with early snow melt on its own without warming, warming on its own, and then a control where the plants aren't manipulated at all. And so this is called a factorial design. So we crossed everything, and then this is all replicated 7 times across the landscape. And so our specific questions were, 1, what are the effects of each treatment on flowering phenology? And 2, does snowmelt timing affect flowering phenology through temperature accumulation, or is snowmelt timing a distinct cue? And I just want to bring this picture up again to show you, you can see here with this little green square, this is an area where the snow is melted early. You can start to see, it's really funny to be out there and see the plants greening up earlier. And you get these little squares in the landscape that are advanced. So what we did was we looked at 3 different plant species. These just happen to be the most abundant ones that we could look at. And they're shown in the order in which they flower. So the first is Delphinium nuttallionum, which is a larkspur. The second one is Valeriana edulis, which is valerian, which you might have heard of in your bedtime tea. It's not the same species, but it's the same genus. And then Potentilla pulcherrima, which is a cinquefoil. And so now I'm going to show you some results. And first, we're going to disentangle the effects of each treatment. And before I reveal all of this, I'm going to walk you through this figure because there's kind of a lot happening. So on the bottom here, we have our 4 treatments: control, early melt, warmed, and the combination of early melt with warming. And on the response, or the y-axis, we have the peak flowering day of year, which is our fancy way of saying date of when they flowered. And so first of all, if we just look here, we see the plants definitely flowered earlier in response to early snowmelt. And this little legend up here is showing you there's a little star next to it. That means this particular comparison was significantly different. We see the same thing for warming as we expected. So we see earlier flowering leading to, warming leading to earlier flowering. And then we can also take a look at this combination treatment. And here we're looking at something a little bit different. We weren't interested in whether it's different from the control. Instead, we were interested in whether the combined effect was, as you might expect, if you add the 2 other treatments together, or if it differed in some way. So sometimes we see synergistic effects when we manipulate 2 things together. So maybe we would see even earlier flowering than expected, or not as big of a change in flowering. So this white dot is the amount, is the expected change just from adding the snowmelt and the warming treatment together. So as you can see, these didn't differ. We call that an additive effect. Okay, so that answers question 1, but what about question 2, the potential mechanism behind early snowmelt? And for that, we had to do this with a statistical analysis. And so what we did in our models, we add growing degree days to the model to account for any effects of growing degree days, and then say, is there still an effect of snowmelt left that we can pick up on? And indeed there was. So after accounting for the effect of growing degree days, we're still seeing that effect of early snowmelt. And so this suggests that snowmelt is acting through some other mechanism besides heat accumulation. So what did we see for the other 2 species? That was Delphinium. For valerian, we saw the only effect was early flowering under early snowmelt conditions. And you can see this in the combined treatment as well, right? It's totally the same. But similar to Delphinium, when we account for heat accumulation or growing degree days, we still see snowmelt acting through growing degree days and through something else. Then what about our cinquefoil? We see, just like the larkspur, flowering is earlier under both early snowmelt conditions and under warmed conditions. And the combined effect did not differ from the additive effect, even though we don't have error bars on these. So it looks like they're far apart. They're not differing significantly from one another. And here, though, we found something a little bit different. There was no effect of growing degree days at all. Snowmelt is only acting through something else in this case. However, we did expect growing degree days to matter because it almost always does for most things. And what this tells us is that our warming treatments are also doing something besides warming the air, which we actually already knew as well. So there are some well-known side effects to these warming chambers, mainly the main one being that they can also increase humidity. So what does this all mean? All the effects on phenology were additive, and this is kind of handy because it means it makes prediction easier. We know that when they happen together, to understand how they're interacting with each other, we don't have to account for any kind of weird synergistic effects. We saw that snowmelt is an important cue of flowering phenology for all of the species. And for all of them, it was acting through some other mechanism other than heat accumulation. So that's pretty cool. And also that the accumulation of temperature was not a phenological driver in all species. So just that one, the potentilla, didn't have that effect. Also, a single climate signal here, snowmelt timing, can have multiple mechanisms for changing phenology. So it's important for heat accumulation. It's probably also acting through soil moisture in some way or another. And that, in fact, is the next step for this work. I'd love to do a project down the line where I'm able to manipulate soil moisture and nutrients and figure out which one or both of those things might be the other effect by which snowmelt is affecting the timing of flowering in these plant species. And this is important, right, to understand exactly how climate change might be affecting all these plants. So that concludes the first part of the talk, where we're thinking about the drivers of phenological shifts, and leads us to the next part where we're going to think about consequences. And we're going to think about those two consequences today. So first, we're going to think about population viability. And before I dive into my specific example, I kind of want to take a step back and provide a little bit more context for this work in particular, and just ask more broadly, what are the demographic consequences of phenological shifts under climate change, broadly speaking? This was a review paper that I led last year, and it was actually really fun to read about all sorts of organisms besides plants and figure out what all's going on. So we had some search criteria that revealed over 1,200 papers that we sorted through to see which ones met some specific criteria. And 238 of those papers met our criteria, which were: one, did the study quantify a phenological shift through time or phenological sensitivity to climate? And what I mean by phenological sensitivity is simply the response of an event to, for example, warmer temperatures. So say plants have advanced flowering by 1 day for every 1 degree increase in temperature, be a phenological sensitivity. And two, did the study also measure a demographic consequence? So we could attempt to link those two things together. And so when we look through all these papers, one thing that jumped out at us is that 78% of the papers on this topic have been published just within the last decade. So it certainly has become a popular question for scientists. So a lot of people are thinking about this question. And of the different demographic consequences, here's how we broke it down. So some of them are called vital rates. This is an important concept that will come back later in the talk. It's parts of the life cycle that influence population growth rate. So here we're thinking about growth, survival, reproduction, and recruitment. And what recruitment is the recruitment of new individuals into a population. So for plants, that would be seedlings. For animals, new babies. Those are called, that part is called recruitment. And then population just refers to any population level consequence. So either the number of individuals or a population growth rate. And so one thing that jumped out to us here is that most papers focus on reproduction, and that's fine. However, a lot of organisms might be affected by other vital rates more so than reproduction. And so I will explain that. And I will also break this figure down. So these are all called life cycle diagrams. And I'm going to start just with a simple one, the annual plants. So here we have an adult plant. It reproduces by making seeds, but then those seeds have to germinate and produce seedlings in order to see recruitment. So that's when we get a new individual into the population. Then that new recruit will grow and survive all within the same year to an adult, and the process starts all over again. So that's our life cycle diagram. You'll see here the arrows, if they're thick, that represents the part of the life cycle that has the biggest effect on population growth rate. And we know just from all kinds of research that has happened in the past, for short-lived organisms, reproduction and recruitment tend to influence population growth quite strongly. And that makes sense if you think about it, right? You have an organism that gets one shot at reproducing in its life. So if it doesn't and it fails, that's likely to have a big effect on the population as a whole. In contrast, longer-lived organisms have multiple chances to reproduce throughout their lifetime. And so here we might expect they could experience quite a bit of reproductive loss or failure in any given year and be okay as long as they're reproducing in other years. So in this case, growth and survival are more important, have bigger effects on population growth rate in long-lived organisms. And so finally, the last thing that stood out to us here is that there's really not much known about the population level consequences of phenological shifts in plants. So only 2 studies, and one of those studies is my own, and I'm gonna talk to you about that next. So this study was done on a plant species called the aspen sunflower, and it's shown here. This is the meadow where we study it, and all these yellow flowers are the aspen sunflower. This is just a little side note, but its flowers always face the east. It's not heliotropic like other sunflowers that follow the sun. This one's just always facing east. I guess if you're lost, it can be handy. So this is what it looks like, and it flowers in mid-July. It's a pretty common and abundant species in these meadows. It flowers earlier when the snowmelt date is earlier, just like many of the other plant species. And it also is experiencing increased frost damage, which is a little bit counterintuitive. You might say, wait, why are we seeing increased frost damage under climate change? And this is one of these counterintuitive effects. So what we have here is a plant species that's now flowering much earlier than it used to in the past. And it's now flowering at a time of year when nighttime temperatures dip below freezing. And it's being exposed to these nighttime temperatures that it's not used to dealing with or adapted to dealing with, and they get frosted. And this is what they look like when they get frosted. They do not go on to make any seeds or flowers in that year, but these are long-lived perennials, so they can try again next year. So this is the meadow where we study this plant and what it looks like when a year when snowmelt is late, contrasted to a year when snowmelt is early. So it can have a huge effect. This picture was taken on the same day in 2 different years. So again, here's our happy sunflowers, and then they all got frosted in another year. So I really wanted to know, how does early snowmelt affect the population dynamics of this species? And if we make our hypotheses, we've already thought about reproduction being decreased by frost damage, but what about growth and survival, right? We have to think about that because this is a long-lived perennial and these things might be really important for this species. So one thing we might expect to see is increased growth under early snowmelt, simply because the growing seasons are longer when the snow melts out early. In contrast, we could make the opposite prediction that we would see decreased growth because we also see longer drought periods when snow melts early in this ecosystem. And that's because we have the monsoon rains in July, and those are not really changing as the climate changes, but we're having snow melt date shift to an earlier date, and that's lengthening that dry period. And for the same reason, we expect to see decreased survival in response to early snow melt because of that drought period. So to address this, I used some long-term demographic data. This project was started well before I arrived at the field station, but I did end up collecting 5 years of the data. We have a series of tagged individuals in our study population. So every individual gets a unique identification number and a little metal tag, and we find that individual every year, measure its size, which is measured as the number of rosettes or above ground meristems. Then we also record how many of the flower stalks were frosted and how many were okay and went on to go on and make seeds. We also very carefully dig around and look, we have to get down on our hands and knees with our faces right on the ground to look for seedlings, and we record how many seedlings are there each year. And this was done over 3 elevations that span the elevation range of this species. So we've got low, middle, and high. I also combined this with a snow removal experiment that I did in 2014. I'm not going to have time to talk about it today, but I wanted to mention it because results from the snow removal experiment are consistent with the long-term observational data. So anytime, you can have that experiment showing the same results as the observational study is a good, provides even more evidence for what you're seeing. So what did we find? First, I'm going to show you, this is the rate of frost damage across the 3 populations. So a snowmelt date on the bottom here, predicting the probability of abortion, and that's just technically what happens. The flower buds get aborted when they get frosted. And so we see early snowmelts associated with an increased probability of abortion in response to frost at every population across the elevation range. And now what about some of our vital rates, our other vital rates not related to reproduction? So these are a little different. We have plant size now on the x-axis. It's been log transformed, so the numbers, that's why the numbers are so small. And then we have plant size in the following year. And I'm just going to show you first a low elevation. First, if you just don't pay attention to the two colors of lines and look at the shape of the line, you can see bigger plants tend to grow more, which is very common. We see this across all kinds of plant species. And then if you look at the colors of the lines, the red line represents early snowmelt. So what you can see here is that when snowmelt is early, the plants do tend to grow more. So what about the other populations? It's a little different for the high elevation. So here, the red line's below the blue line, meaning that we see decreased growth in response to early snowmelt in this population. We don't know exactly why that's happening, but we think it's probably related to the fact that the soils are a lot shallower at this elevation and more rocky. And so there might be a bigger effect of drought there. It might be harder for those plants to take advantage of soil moisture. And then what about survival? Last but not least, this effect is consistent across the populations. It's reduced when snowmelt is early. And we think this is probably a drought effect. We did a little pilot watering study, and the watered plants did survive better. So it's consistent with this idea that drought might lead to decreased survival. But what we really are after is, how does this affect population growth rate? So if we put all these vital rate equations into our population model, what do we find? First, I'm just going to show you the low elevation population. So now we've got snowmelt date on our x-axis and lambda on our y-axis. And this is just a measure of population growth. A lambda of 1 means the population is stable, so it's neither increasing nor decreasing. So values below 1 means the population is declining, and above 1 it's growing. And these are, this gray shaded area is called a 95% credible interval, and it's just showing some error around our mean line of fit. And when that gray line does not overlap with 1, that's really good evidence that the population is declining. So we see this trend toward population decline under early snowmelt conditions. And what do we see for the other two populations? Similar trend, except that this population is declining much less, and the high elevation population is actually declining much more. And we'll figure out why in a moment. Well, you might already be thinking why. So the reason I really love building these kinds of population models that I build is that you can figure out why a population growth rate is changing. So it's not our goal to say, like, this is the exact growth rate of the population, but rather what part of the life cycle is being affected to cause this either increase or decrease in population growth. So the way you figure that out is called a life table response experiment, for those of you in the audience that want to know about that. And it gives us results that look like this. So now we still have snowmelt date on our x-axis, but we have the contribution of each vital rate to the response of lambda to snowmelt on the y. And I know that's a mouthful, but it's really, so basically the way to think of this is that a positive value here means that later is good for growth, and a negative value means that early is good for growth. So here, the purple line is survival. Later is good for population growth. So that means early is bad, right? And this is what you can, because this is the largest effect, that tells us that the main reason we're seeing a decline in population growth under early snowmelt conditions is because of decreased survival. So we're seeing a small effect from that frost damage as well, shown by the red line, but mostly the survival. So what about the other two populations? Over here for the mid elevation, they're kind of equal. So the frost damage and survival are both contributing to that declining trend. But remember, that was a very weak trend. It was mostly stable. And in the high elevation, we see survival and also green here for growth. And if you remember, that's the population where growth was responding negatively to early snowmelts. And so this is really cool from a biological perspective, I think, because it was a tiny effect. If you remember, the red line was barely below that blue line, but it can have a huge effect on population growth rate because the population growth rate is really sensitive to changes in growth and survival in this species. And so we started out this particular section by by asking, how does early snowmelt affect population dynamics? And despite this really large and visible, obvious effect of frost damage, because of in response to earlier flowering, we see that's not the primary way that snowmelt reduces population growth rate. It's mostly via survival. And this is consistent with what we know about the life history of the species and long-lived species in general, right? We expect survival and growth to have the biggest effect. But it's really nice to be able to put them all together and to a population model to see what exactly is going on. So what is next for this work? I want to go back to this slide. We see here, again, calling out that there's really little work in plants in particular on the consequences of earlier life history events. And my study and the other study both got at this indirectly. So I didn't directly model the effects of earlier flowering on population growth, right? I included a consequence of earlier flowering, in this case, frost damage, to see how that affects population growth. But I really love to do some direct manipulations of phenology, and that's exactly what we're doing now, using these snow tarps that I mentioned earlier. So here's an actual picture of the tarp before the snow is melted out, and we're manipulating the snow to say, how do early life cycle events affect population growth? And this is a drone, a photo taken from drones. You can see the early squares melted out early in the landscape. So last year we started doing this for the first time. And I currently have a proposal pending at the National Science Foundation. So if I get money from them, I can do this project and build population models that directly incorporate timing into them. And I'd love to do it for multiple plant species. But in the meantime, I have a PhD student who's able to work in these plots and just start to understand how do we see these drought effects in other species, and is it affecting plant physiology in a consistent way with what we're seeing with the other vital rates? So far, yes. I don't have time to go into her work today, but she's done one study, one field season so far, and I'm excited to see what she finds this year. So that brings me to the last part of the talk, where we're going to look at one other consequence. And don't worry, this part of the talk isn't as long because I know that there are time constraints. And so we're thinking about interactions for pollinators. So typically we might think about plants competing directly with each other for water and soil nutrients and things like that, but they also compete with each other for pollinators. So you can think of pollinators as a resource in this case. And what we see is when plant species are flowering with each other and their flowering periods overlap, they're more likely to compete for pollinators. And so I just want to go back to this highlight this variation in the species level changes that we're seeing in the timing of flowering, to say that this all results in kind of a reshuffling of who's overlapping with whom in the community. And this can be seen here in this figure that I'm going to show in just a second. So any green dot squares mean there's a pair of species that is overlapping more now than they used to in the past. And a blue square means it's a pair of species that's overlapping less than they used to. So I don't want you to try to look at the squares in particular, but just notice there's quite a few green and blue squares in this figure, showing that the co-flowering patterns have changed. And so I really wanted to know, okay, what are the consequences of these changes in flowering timing, in particular of altered co-flowering? And so what I did with one of my master's students was pick out a pair of species that is changing in the data set to exactly get at this question. We also were interested in one other thing, especially in the context of these drought effects that seem to be popping up and being important in a lot of other work as well as my own, but knowing that the biotic interactions may also be affected by abiotic conditions. And we know much less about how these two things might interact as the climate changes. So this is work that my master's student Marie Faust performed a few years ago, and also wanted to give her a shout out. I'm excited to talk about her. This was just published this year. So we have two species that we focused on: Linum lewisii, the common name is blue flax, and our friend Potentilla again that we saw earlier. And this is the focal species, flax. We're really going to focus in here on what are the consequences for this species. And the reason we focused on this one and not that one is because flax depends on pollinators for reproduction, whereas potentilla can is able of self-pollinating. So we know from the long-term data set that this pair of species has decreased in co-flowering by 25%. So at the top here, we can see this is the first decade of the long-term data. Potentilla kind of totally overwhelmed linum in its flowering. And in the most recent decades, it's not the same. We see that linum has shifted forward in time, and there's some days when there's linum flowers without any potentilla flowers whatsoever. So when we talk about co-flowering, it is about the number of days that they're overlapping with each other, but also their relative floral abundance gets taken into account as well. So in this project, we wanted to ask if shifts in co-flowering affect interspecific competition for pollinator services between these two species. And we expect that less overlap will lead to more visits and more pollen deposition. And if we ask the same thing about reproduction, we expect that if this holds and we see changes in pollinator visitation, we would expect to see more fruits and seeds as the species overlap with each other less. And then finally, we're interested in this effect of drought. And we were able to study this because it was a dry year by adding water. So we have, does this effect maybe only happen when it's wet or only when it's dry? So the way we got at this is called a randomized split plot block design, which is kind of a mouthful. But we have 3 treatments where we have our control where nothing has changed, a treatment where we removed half of the potentilla flowers, and then another where we removed all of the potentilla flowers. And in this way, we can create this experimental gradient of co-flowering. And the split plot part means these plots were split in half, and that half of the linum got additional pollen added to them. And the reason we did that is because we can understand if changes in pollination are likely to be a problem for the plant species, because sometimes they get plenty of pollination and getting more isn't necessarily better. So in this case, we had each treatment separated by 4 meters and grouped together. So this is called a block. And each block was separated by 12 meters, and we replicated this 12 times across the meadow. And then half of them got water. And Marie spent a lot of time watching the plants and recording who came to visit. We also had an undergraduate student who contributed a lot to this effort as well. And then we dyed the pollen grains and counted them and counted fruits and seeds in the lab. So the first thing to look at is, did our treatments work? And yes, indeed, watering did increase soil moisture by 3%, which might not seem like a lot, but to a plant, that's a lot. And then if we first look at visitation, now we've got co-flowering overlap here shown as a value from 0 to 1. So 1 means they're totally overlapping with each other, and 0 means not at all. This is just a bunch of scatter, right? There's actually nothing happening. It did not affect pollinator visitation rates or the number of visitors. What about pollen deposition though? We did see one effect in terms of pollen deposition. So on the bottom, we have the number of pollen grains. That didn't vary. However, the proportion of linum pollen grains did, and we call that pollen quality. So what we're seeing here is that when there's more co-flowering overlap, there's a lower proportion of linum pollen grains. And that's because the plants are tending to get other pollen species deposited on their stigmas when they're overlapping more with potentilla. Okay, did any of this affect seed production? So here we're showing total number of seeds, and it's broken out by the control and the water treatment because it did depend on whether they were watered or not. So we see here, the total seed production decreased with more co-flowering, but only in plants that were watered. And there is a lot going on in this figure. We have plant size in here because the larger plants made more seeds than the smaller plants, and we had to account for that in our model. But if you just sort of pay attention to the overall pattern, here you can see this trend of decreasing seed production with increasing co-flowering. And additionally, we didn't find any evidence of pollen limitation from our hand pollinations. Instead, we only saw seed production increase with water addition, suggesting that reproduction is more affected by water than pollination in this case. So I want to show you this awesome graphic that my student Marie made, what we think is going on here. So when they're overlapping with each other, this is the case as a bumblebee, but we saw a lot of flies pollinating these species as well. We have a bee who's more likely to move from one flower to the next when they're foraging. However, when there's less overlap, in this case, no overlap, they're more likely to go from one line of flower to the next, and in that way transfer the right kind of pollen, or we call that conspecific pollen. So here, only getting linum pollen. Up here, also getting potentilla pollen. And in the observations, we did see the flies and the bees moving in between these plant species quite a bit. And then this did indeed affect seed production, but only in the plants that were watered. So only when the plants didn't have this limitation, have the reproduction limited by water availability, or they able to take advantage of the kind of better, higher quality pollen deposition and make more seeds. So what does this mean for linum? It could mean that climate change might have a positive effect on linum reproduction via these decreases in co-flowering. So it's getting pulled apart in time with potentilla, and maybe that's good for its reproduction. However, these abiotic changes will counteract this positive effect in dry years. So it's really more about, from an overall perspective, what is the abiotic environment looking like? In this case, the drought context, which is really important because climate change is projected to increase water stress in Colorado and in the southwestern US in general. We also found in this work that biotic and abiotic effects of climate change can interact with each other. So that's that part where, yeah, the pollinators matter, but only when it's not super dry. And so to conclude then, this is a photo that my friend Jane took from the area, just up valley from the field station. You know, a lot of work from the Rocky Mountain Biological Lab is pointing to the importance of drought effects under climate change. Here, this is what seemed to affect our Helianthella in terms of its population viability. We saw drought mediating the effects of altered pollination, and also soil moisture in some way playing a role in the cueing of phenological events. So if there's one thing I want you to take away from this talk today, is that as the climate continues to change in this area, drought is probably going to be very important for ecological timing, for the consequences of shifts in phenology, and for a lot of other processes as well. So with that, I'd like to thank all of my awesome students and my collaborators and my funding agencies, and I'm, of course, happy to take questions.
Thank you so much for that wonderful talk, Amy. Folks, if you have any questions for Amy, please pop them in the Q&A box right now. Amy, it was so good to see your work at the Rocky Mountains. It was really like a lovely throwback for me when I used to work there. And it's really quite apparent how much more complex questions you can ask when you already have this wealth of historical information and for a given location. And yeah, I really like that photo you posted with the sunflowers in the year without the frost. And then in the year where they got frost, that is very dramatic. Like they're just like no flowers at all.
It's so dramatic. And so you think like, this must affect the population. And then it's fascinating that like, yeah, I mean, it does a little bit, but not as much as you would guess. Yeah, and that's the other thing. I think that graph you showed of the comparison of, I suppose, the relative importance of vital rates to the growth rate. That is a really cool graph and really cool results. How long do these sunflowers live for? Oh, great question. We don't, the short answer is we don't know. But at least, now the study's been going on for almost 25 years, and there's many, many plants that have been around for the whole time. So we think David, in a way, my former postdoc advisor who started that project, thinks they can live up to 80 years old. Oh, wow. Based on like looking at what may or may not be growth rings. So take that with a grain of salt. Okay. It's really hard to know how old a perennial plant like that is or not. Especially when they don't. I'm sure. Yeah, that's really cool. And when you look at the survival of these plants, was this like death all the way to the roots, or was it just like they're gone? They don't come back. That's very sad for something that lives for potentially decades. I know. Yeah.
We have one question from Anna Wingarden. Awesome talk, Amy. Thank you. I know the Helianthella data set is decades long, but how many years of data are required to build a robust vital rate model?
Oh, that's a really good question. Yeah.
Big question.
Yeah. So you can build, these are called individual-based population models or matrix population models, basically. The fancy new version is called an integral projection model, which means we're using all the continuous size variation. And you essentially need at least 4 years of data, I would say, to get a good handle, a decent handle on whether the population's like increasing or decreasing. If you think about it, 4 years of data means you've got 3 transitions from like 1 year to the next.
Right.
So, and then if you want to do, I kind of glossed over this, but we had stochastic population growth rates, which means we're also accounting for just environmental stochasticity, random variation that can affect populations a lot. And for those, it seems like you need at least 8 to 10 years of data to get those kinds of models going.
And you would need quite a large population size too, right?
Yeah. That is actually trickier. At least, 1 to 200 individuals minimum. This one
just 1 to 200.
I know, right? These data sets have like, I forget. I think the lowest one is like 450 individuals. Yeah.
We have another question from Evelina. They first want to thank you and say that's wonderful information. But they also ask, how does this process affect honeybees, and will some types of honey go extinct?
Oh, interesting. Yeah, people always want to know about the honeybees. They're very important agricultural species. And actually, we do not have any honeybees at this elevation. They can't overwinter. It's too cold, so the colonies can't make a go of it, basically. And I also want to point out honeybees are not native to North America, so they're actually a European species. And so, they're really important for agriculture, like I said, and we think of them as, so that's kind of like a management problem, whereas our native bees that are declining, like bumblebees or sweat bees, maybe if you've seen those or heard of them, are more of a conservation issue. And that is a great question to know about how some of this stuff might affect the bees. And, we don't, the cop-out answer is we don't know for sure, but we're seeing, there's lots of ways that it could matter. So altered timing, there's this idea of phenological mismatch where, the plants are not responding to the same cues as the bees, then they might be coming out at different times. And I know Lloyd has done some work on this question, actually. And I think largely what we're seeing, and this is coming from the review paper that I wrote too, is that bees, in terms of timing, unless you're at the very beginning of the spring coming out kind of the first thing, oftentimes they are generalists and they will have other plant species that they can get their pollen and nectar from.
Right, they're quite flexible.
And they're pretty flexible, yeah. So the species that are coming out really early that might happen to emerge before there's any flowers, that could be a problem, or if they're specialized, that can be more important for timing wise.
Those tend to be the natives that are the specialists, not so much the honeybees, which are
honeybees are definitely generalized, and bumblebees as well are quite generalized.
Right.
Yeah.
Okay, so honey is safe.
Honey is, oh yeah, I forgot about the honey part of the question. Yeah. In terms of all of this stuff, it's more important what's going on with their kind of agricultural species probably. But we do see the frost damage happening in fruit trees that flower in the spring, and it can have a big effect on yields like cherries or peaches or apricots or things like that.
Okay. We have another question by Shauna, and they ask, is it possible that survival, I assume, of the plants is affected by getting washed away in snowmelt?
Oh, yeah. Well, we haven't seen that yet. So they're kind of used to the snow melting, although it is, the snow is melting at a faster rate now than it used to as well. So, maybe the runoff could be stronger, but it's, no, we're not seeing anything like that happening. It's more just an issue that there's not as much water content going into the soil from the snow, or it's going in earlier. Yeah.
Presumably that affects hydrology in the streams and
totally.
stuff, the aquatic animals and plants.
It does. Yeah. I mean, it's really amazing. Eloy has seen this as well. When the snow melts in the spring, the streams are just gushing with water, and there's just so much water coming off the mountain. But these plants aren't growing right by the streams, or they're kind of more away from it. Yeah.
We have another question by Evelina, and they're asking about their hydrangeas, and they have been filled with leaves twice this year, but then those leaves got killed off because of frost.
Oh.
So they're asking if their hydrangeas will bloom less this year. What have you learned from Helianthus' work?
Yeah. So the leaves, I think it seems like the leaves have been killed by frost, is what you're saying.
Yeah. And will that affect the flowering?
Will it affect the flowering? It might. It could, because that's the leaves are the way that the plants do photosynthesis, and that's how they make their food and get resources. So think of it as kind of like a bank account, right? They got to have enough to spend the resources on flowering. But they might also have enough kind of stored resources below ground that they would be able to still make flowers. But you should look and see, and make sure you take note if there's fewer flowers this year.
Yeah. Science at home.
Yeah, science at home.
Well, thank you so much, Amy, again, for this wonderful talk and for answering so many of our audience questions. It's almost 7 o'clock now. So thank you, everyone who's at home, who's attending, and wherever you may be. Thank you for joining and participating in Science Cafe. If you want to watch this talk in the future, remember you can check out this URL, which I will post on the screen. If I can figure out the technology. There we go. Yeah, so check out this URL if you want to rewatch this. Next month, we will have our, Oh yeah, I was going to say that if you want to check, if you want to search for this URL, it might be easier to just go online, just do an online search for ABG Science Cafe, and you'll find all our past recordings up there. And you can also register for next week's talks. Next month, we will have our first in-person Science Cafe in a long time. We're very excited. And as always, it'll be on the third Thursday of the month. That's May 19th, 6 p.m. Eastern Time. The location will be Day Hall at the Atlanta Botanical Gardens Midtown location. We'll be hosting Georgian Eubanks, a writer from the University of North Carolina Press, and their talk will be titled Saving the Wild South, the Fight for Native Plants on the Brink of Extinction. As always, we welcome questions and suggestions. Send those to Ian Sabo, our field biologist, who also helps to coordinate Science Cafe. Thank you very much, Ian. You can reach Ian at isabo@atlantabg.org. You check out our website as well for more information on our conservation and research work. It's atlantabg.org/conservation-research. Don't forget to follow us on Instagram, where we post cool plant photos and our latest news on our department's conservation and research activities. Our handle is @AtlantaBG Thank you, everyone, for being here today, and have a lovely rest of your evening.
Thanks again.